Well, hello everybody. Welcome. Good day to everyone, and thank you so much for joining us via Zoom for the Covestro Lectureship in Sustainability. My name is Mark Hillmeyer. I'm a faculty member in the Department of Chemistry here at the University of Minnesota, and I'm also the director of the Center for Sustainable Polymers, the NSF Center for Sustainable Polymers, which is very happy to co-host this lectureship. Let me just say a word about the Covestro Lectureship in Sustainability. Sustainable, this is from their website, sustainable thinking and acting are essential to preserve our planet, improve both the basis of life and the quality of living for millions. We align economic growth with sustainability, with profitable products and technologies that can benefit society and reduce our impact on nature. As I think a sign of their commitment, Covestro is now one of more than 45 global companies, and they ended up being also a co-founder of the Alliance to End Plastic Waste, which is a nonprofit organization dedicated to developing, deploying, and bringing to scale solutions to reduce, reuse, and recover plastic waste. So I think Covestro has had a longstanding commitment to sustainability. As part of that commitment, and a sign of that is that they have been a wonderful sponsor of this lectureship now since 2012. Recently, we've had talks from preeminent figures in the field, such as Richard Thompson, Jillian Buriak, and Karen Goldberg. I want to give a special thanks to Don Wardius from Covestro, who has been our principal contact to support of this lectureship over the years. It's also important for me to thank the team at the Center for Sustainable Polymers, Jennifer McCambridge, Laura Seifert, and John Beamer, who in now pandemic times have transitioned from organizing a big lecture hall at the University of Minnesota, which is also not easy, to organizing a big Zoom webinar event. And I'm so happy to see the number of participants who have been able to come. We have had snowstorms during this event, so we can at least be in the comfort of our own homes and offices. And if you were in Minneapolis today, you might have to wear a jacket, but it's a lovely day. Before I introduce our speaker, I do want to say just a little bit of housekeeping. If you're having any technical problems, you can use the chat function, and maybe we can help you sort out any technical problems. But throughout the entire lecture today, please feel free to use and encourage you to use the Q&A function in Zoom to ask questions. So in the next few slides, if you see a question already you'd like to ask, please just send it. I'll moderate all the Q&A at the end. And you can ask at the end as well. But if you ask during the lecture, it'll help me kind of organize a little bit some of the initial questions that I can ask. So I will now turn to the introduction of our Covestro lecturer for this lecture in sustainability for this year. That's Professor Kris McNeill from the ETH in Zurich. Hello, Kris. Thank you so much for doing this. Very good to see you, and thank you for accommodating this. And Kris earned his degree, undergraduate degree from Reed College, went to earn a PhD from the University of California, Berkeley, did postdoctoral work at MIT. Kris and I have a long history together. Kris joined the faculty at the University of Minnesota in 2000 and was on the faculty with me from 2000 to 2009. And, 9 years with me is pretty much enough for most people, Kris, and I can understand what happened there. But you moved to join the faculty of ETH Zurich in 2009. In short, so we can get to his lecture here, Kris has worked on a number of very important environmental problems associated with environmental chemistry, and has really been an international leader in the field without question. He has also had a tremendous passion for teaching, and I can remember really working with him early on in his time here, talking about the best ways to effectively teach physical organic students the concepts of thermodynamics, and really getting into it. And that passion really comes through. One short story is that there was a billboard in Minneapolis about, please come to a local college where the professors know your name. And Professor McNeill was teaching a general chemistry course at that time with over probably around 300 people of it. He didn't like that. He thought you should come to the University of Minnesota. So he decided to memorize everybody's name in the class. And he would see people on campus and call them out. They were terrified. They had no idea that the professor knew their name. But I thought that was a real testament to his passion to engaging with students. And that's very clear in his coursework delivery. And I'm sure we're in for a real treat today with this lecture. So, Kris, I'm going to turn it over to you, and looking very much forward to your lecture. Thank you so much.
Thank you, Mark. Thank you. I was thinking when you were talking about how I first met you, which was that you were my host, my student host, when I was a prospective grad student at Caltech. I didn't end up going to Caltech, so you know how well that went. Just teasing. No, but it was true. I did meet Mark then, and then I was really, really lucky to have him as a faculty member because he took a lot as a senior faculty member because he took a lot of interest in helping the junior faculty, and I learned so much from him. And I mean, one of the funnier things was he was like, "Hey, I'd like to learn more about physical organic chemistry. I'd like to come sit in on your course." And I was just terrified because, of course, teaching a class for the first time is already terrifying. And then having a professor sit in the front row was terrifying. But it made it so much better. And I'm really, really thankful for you for doing that. Anyway, thank you for the invitation. And maybe I'll just, we don't have so much time. I should probably start talking about the actual talk here. This is an environmental chemist view of biodegradable plastics. Actually, this title screen's been up so long, I noticed that the Chemists is the larger font size and everything else, which is actually a mistake, but it's kind of a cool one. Oh, geez. Am I going to have fits? Okay, there we go. It's kind of a cool one because I actually think that's one of the things that I feel like is my value added, if you will, to the environmental science community is that I come from an organic chemistry background. And I think I have a slightly different perspective on some of the problems than some of the other folks. And I hope you see a little bit of that today. So maybe we can talk a bit about environmental organic chemistry, kind of where it's been. And as you'll see, it's a bit of a repeating story. So I'd say the beginning is really DDT. So DDT is this famous pesticide that was developed by Paul Hermann Müller in Switzerland, actually, in the kind of time of World War II. And following World War II, the DDT production went, kind of crazy. And then Silent Spring was written, and the enthusiasm for DDT went down, and so did the production. And that is kind of, there's sort of a boom and bust. And what's interesting is you can actually see that in the sedimentary record. You can go to this lake near Dallas, Texas, as the USGS did, and you can collect, so lakes, you lay down sediment in layers, and you can actually collect those layers if you push like a plastic or glass tube into the sediment and haul it out carefully. And then you can go through and look in each of those layers. It takes a little work to figure out what year they correspond to, but you can make a record of how much DDT was accumulating in the sediment at each year in the record. What you see is the record of DDT matches the production of DDT almost exactly. It's kind of remarkable. And what's also a theme is that there was this enthusiasm for the chemical, there was some concern about the chemical and lack of enthusiasm, and then it was, it faded out. And that whole time scale is about 40 years. And we see that again and again and again. This is a different molecule. These are, well, actually, it's a stand-in for 209 molecules. It's one of the PCBs, and this one's 99, I think. And the PCBs, sort of follow a similar trajectory. Growth and decay on a time scale of something like 4 decades. And it doesn't matter if it's an agricultural chemical or or if it's an industrial chemical or even a pharmaceutical or a personal care product, we see the same trend, that there's this growth of use and then this decay. And it's been kind of depressing for us environmental chemists. What we say is, if someone would have just asked us at the beginning, before they decided to make thousands of tons of this molecule, about its environmental behavior, maybe we could have saved some bald eagles or something. So we feel like we're at the end of the pipe, and we keep like complaining that nobody asked us at the beginning of the pipe. Right. So today, to introduce the talk, I'm going to give an introduction I've given many times for a different talk, actually, not this talk. And it occurred to me while I was writing this one that it's strangely relevant. So what I've talked about in the past are how PCBs are like rubber ducks. And I use this as an example of how, basically to give you a sense of what motivates me in environmental chemistry. And so the story goes like this. In January '92, there was a boat, a big cargo ship going from Hong Kong to Tacoma, Washington, and it was carrying plastic bath toys. And somewhere along the way, in the middle of the Pacific, there was a storm, and some of these cargo containers containing plastic ducks and plastic frogs and beavers and turtles washed overboard. And, about approximately 30,000 of these bath toys ended up in the ocean. Then they started washing up. Sometime later in the year, they started washing up on various shores. Notably, a big concentration of them landed in Alaska. And this kind of got the attention of a beachcomber network, and some citizen science started happening, like, oh, maybe we can understand something about how the ocean currents work by collecting all this data. And so there was this whole initiative to document all the sightings of these. What's pretty cool is they kept this going for a while, and what they found was that the ducks would return every 3 years. They would come back to the shores of Alaska, and it actually ended up being one of the first experimental estimates of, and one of the best experimental estimates of the subpolar gyre circulation. So this is the subpolar gyre here, and the circulation time was, we had ways of kind of guessing at it, but no one had ever dumped 30,000 plastic, drones into the water and just let them go around. I mean, can you imagine the NSF grant proposal to do that? First of all, it would never go through the ethics clearance. Second of all, you can imagine what reviewer 2 would say about, like, yeah, how are they ever going to hope to get enough ducks to rebuild the, blah, 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 blah. But the point is that we do these kinds of things all the time. Society dumps stuff into the environment. Sometimes it's a spill like this. Sometimes it's non-point. There's all kinds of ways things get into the environment, contaminants get into the environment. And me as an environmental chemist, one of the things that excites me is that following these ducks, you can learn something about how the world works, how the environment works, And I always did this as a metaphor, like the ducks are like molecules. But in this case today, we're going to talk about the ducks. We're going to talk about plastic. We're going to talk about actual plastic pollution. But there will be some, as you'll see, they're linked. The organic chemistry and the plastics are, of course, intimately linked. Before I get too far, let me go ahead and say a word about the team that did this work. It's been going on for 7 years. We've had, there's 3 of us who are sort of at the top of the leadership structure, and then we've how the people actually did the work. So far it's been 6 folks over these 7 years. Some you might recognize, like Melissa Maurer-Jones, graduated from the University of Minnesota from Christy Haynes Group with a PhD, and came to us as kind of one of the people who really got this project off the ground. But what I really want to say here is that, oh my gosh, my phone is ringing. That's something that doesn't happen in the lecture hall normally. So what I really want to say is that Michael Sander is the guy who really should be giving this talk. He's the guy who wrote the proposal, he got the money, he hired the students, he did everything. He's the conductor of the orchestra, and these are like the first violin and the brass and all that. And I'm kind of the guy at the triangle in the back who is very enthusiastic and very happy to be part of the team. And I can't believe that I'm the one who gets to talk to you about it today. But the way the story goes is basically we got a call one day, and it was a chemical company. It was BASF, and they said, we're thinking about making 1,000 tons or several thousand tons of a product that is going to go into the environment. And we wanted to ask some environmental chemists before we did it. And we were like, "You're kidding! This is our dream. This is what we always talk about." And so since then we've been working with them and trying to understand the behavior of biodegradable polymers. in the environment. So I'm not going to motivate the problem too much. I think this crowd probably knows that there's a lot of plastic pollution in the world. I'll just say one thing, just motivate it with like the growth of plastics is just been, it's been bananas. It's exponential. There are little dips in different financial crises, oil crunch, or maybe like a global financial crisis. But in general, you can see where this curve is going. We also know that there are some environmental effects. This is a pretty famous picture. You may have seen it before. I feel like I see it all the time, but then again, it might be from the talks that I go to. This is a picture taken of a dead albatross at Midway, which is, like halfway between the west coast of the U.S. and Japan. And in fact, that's why it was an air base for World War II. And what you can see here is in this carcass, you can see a lot of plastic that the albatross ate, and it just doesn't look good. What's interesting is that this picture has been taken before. Actually, in 1966, about 100 miles away at this southeast island of Pearl and Hermes Reef, which is going towards Hawaii, about 160 kilometers. In this paper here, you can see contents of the body cavity of a young Laysan albatross, similar to many found on the island. And when they lifted off the top part of the carcass, they saw all this plastic material inside. So this is not a new problem. It's it's been going on since the 60s, since sort of over here in the timeline of plastics. But you can just see from this curve that it's obviously going to be an intensifying problem. This is not going to be getting any better. It's only going to be getting worse. So what do we do about it? Well, the fact is we know what we should do in general. We know that we should probably, reuse, reduce, recycle. Or if you read B. Johnson's book, refuse, reduce, reuse, recycle, and rot. That's like compost. Or if you've been thinking about it in terms of a circular economy frame, you think about we produce stuff and then we try to keep it going as long as possible before it goes to waste. And all of that is very sensible and is exactly what we should do. Another way to think about it is if we're worried about plastics in the open environment, we have to recognize that they're a balance. What's in the open environment is a balance of what's produced, the rate at which it's released, and the rate at which it's being degraded. And right now, the rate at which it's being released for most plastics is much higher than the rate at which they're being degraded. And so this reservoir is going to grow and grow and grow. And so, frankly, just between you and me, the 211 of you and me, the growth part of this is what we really should be working on. But I'm not going to talk about that. I'm going to talk about this part because this is where the science, the interesting science is. Over here, that's also interesting social science. That's like psychology and economics and maybe some sociology and political science and all kinds of stuff is over here. But actually, the chemistry aspects are pretty interesting on this side. So we're going to talk about the blue arrow today. But I want you to know that if you really want to solve the problem, you also have to work on the purple arrow. So let me just back up for a sec. Does it even make sense to think about increasing this degradation rate? In other words, could we make plastics that are, say, biodegradable? And is that even a viable thing to consider? And I think that there are some categories of plastics where the answer is no. Like you don't necessarily want the plastic parts in your car or your house, the siding of your house or something to be biodegradable. But there are other applications where I think it makes a lot of sense. And so these are just 3 examples: fishing, agriculture, fireworks. Not all equally important, by the way, but anyway, what they all share is they all use plastic in large amounts, and they're delivered directly to the environment. The plastics are in the open environment immediately. It's not like Tupperware in your kitchen that eventually finds its way into the environment. In this case, they're used directly in the environment. And in each case, they can be difficult to recover, but for different reasons. It might be because the ocean is large, or it might be because you've strapped them to some explosives and shot them into the air. Or it could be that you use very thin plastic on a big field, and then that ends up being a big problem. And so in these cases, it would be nice if you had some programmed degradation pathway or some, not super persistent plastic material. So I think it does make sense to think about biodegradable plastics in some applications. And we're going to focus today on this middle one, agriculture. So what's the strategy? So the chemical strategy would be to have and so polymer is a chain, right? And so you want to have parts of that chain that are easy to break. And the classic way to do this, or the most common way to try this, is to use ester linkages. Ester linkages are, of course, the condensation of carboxylic acids and alcohols. You get an ester and an equivalent of water. And if, you might remember from high school chemistry that if you, in principle, you could push this equilibrium back the other way. If you add enough water, you could push it the other direction. And the thermodynamics certainly support that. The problem, or the good news and the bad news, is that it's kind of slow at neutral pH on kind of the time scales we're interested in. But there's a lot of catalysts. Nature has been developing catalysts for hydrolyzing esters for a long time, and this is something we're going to take advantage of in this strategy. By the way, you could also use polyurethanes in this, any kind of hydrolyzable group. So if you're a polymer chemist, one way you could approach this is you could attach an alcohol at the omega, or the end position of a carboxylic acid molecule, and link them together and make a polyester in this way. And this is like polylactide is like this, or caprolactone, or some of these other polyesters you may have heard of. The ones I'll be talking about today are more of the second family, where you have a diacid and a diol, and you link them together in this way, so that every other unit is a diol and every other unit is a diacid. But it's still, every linkage is an ester. It's still a polyester. And actually, one of the polymers we're going to talk about today, you take two different diacids and one diol, and you mix them together, and you get the same type of polymer where every other one is a diol and every other one is a diacid, but the diacids are randomly mixed between, say, the T, whatever T is, and whatever A is, which we'll learn about those later. And you can play this game, and there's a kind of an infinite game you can play now, even with this very, very simple strategy, which is kind of exciting. It gives you a lot of room to play, allows you to test lots of different materials for favorable properties. Okay, so now we have a strategy. So you're the chemist, you're in the lab, and you're like, okay, so got it, boss. I need to make a biodegradable material. So then you go and like, oh, what does biodegradable even mean? And I'm here to tell you that biodegradable is not a very useful word. Because if you say the banana is biodegradable, it sounds like a property of the banana. It sounds like you're saying the banana is yellow. And in fact, biodegradability is both a material property and an environmental system property. You need both. It's a marriage. It's a partnership between the system and the material. So like, for example, you would not be surprised if I told you that if you put bananas in the Icelandic winter, they don't degrade very quickly. And why is that? It seems obvious, but just let's think it through. It's because it's very cold, and temperature has a big impact, and there's not enough microbes. So you need microbes, and they need to have a warm enough environment to actually get something done. So that's that's a good example then of saying you need to have a match between the material and the environment if you're going to observe biodegradation. These are some other examples that you may have come across. This is Amazon sells these bags. Glad sells these trash bags. This one is labeled biodegradable, which I already kind of told you that drives me crazy because it doesn't mean anything. Not only does it not mean anything sort of scientifically, because they haven't defined where they have the material, but they haven't told you where it's going to be biodegradable. But the other thing is it actually doesn't mean anything from a policy standpoint. There's no biodegradability, standard per se. This packaging is a lot better. They say compostable. And compostable means something. There's actually a standard for compostability. And in fact, this packaging is really good because it even specifies that it's for municipal programs. You might not know this. I didn't know this. I learned it later. But what that means is that these are certified to degrade under industrial compost conditions. Industrial compost is not something you can achieve in your home garden, typically, unless you've got a really impressive compost pile, because the temperature is 50, 60 degrees Celsius in these industrial composters. And the conditions that they are to meet the certification, it's 12 weeks to disintegrate to microplastic, which isn't great, but anyway, that's one of the requirements. It has to disintegrate within 12 weeks, and then it has 6 months to be degraded to carbon dioxide. Of course, there's issues there, and we'll talk a little bit about that. Not everything can be carbon dioxide. There's got to be some other product. Otherwise, these microbes aren't growing. They're just burning it. But so you have to have some threshold, and that threshold is set relative to cellulose typically. But like for home compost, there's no standard at all. And so if you try to put this in your home compost, it probably wouldn't work very well. Actually, I don't want to ruin your day if you're using these. I use these. But if you talk to the people at the recycling, at the industrial compost centers, they actually remove these bags because they don't want to wait 6 months for it to degrade or 12 weeks to disintegrate. All the other stuff inside this bag is going to disintegrate or compost on the time scale of 3-4 weeks or something like that. So they don't they actually remove the bags in Switzerland from the compost. I still use them, but anyway. So here's the trend that I wanted to put in your brain, that if you're going to look for environments where things are going to degrade quickly, look for compost because it's warm and there's lots of microorganisms. Where are you going to find slow degradation? Well, go to the open ocean. It's very cold, often close to freezing, like 4 degrees Celsius would be a very typical ocean temperature. And there's very, very few microbes. I mean, there's microbes for sure, you can find them, but just relatively speaking, it's pretty dilute. Okay, so now you've got your chemical strategy. You know what biodegradation means or doesn't mean. Now you're ready to test your material. What do you want to do? Well, I would say there's 3 things you need to know. There's 3 steps that you need to think about in terms of degradation of a polymer. There's step 1 is the colonization of the material by microbes. You put in a pristine material, the microbes have to find it. And you'll see in this talk that we're going to talk a lot about fungi and unicellular organisms, probably bacteria, but we can't really tell from the techniques we use if they're archaea or bacteria. And then after the colonization, the polymer chains are too large to get into the cell membranes of these microbes, and so they have to excrete something to break down the polymer, and they typically excrete extracellular enzymes, esterases, cutinases, hydrolases. That breaks down the polymer. The small pieces can now diffuse in. Then they can go to work and use that for energy, burn it for energy and make CO2, just like we do with our food, or they could build biomass, and that's also an option. And the relative amount of biomass you make to CO2 you produce would be something that you might see as the growth efficiency of the microbes. So some microbes are much more efficient, they can grow really well, and some are less efficient, but generally it's a balance between these 2 processes. And I've drawn it as a linear sequence, but it's actually, of course, a feedback loop. Like the more you grow, then the more you've colonized, and the more you can grow, and the more you can colonize, etc. But we'll think about it as this chain for now. So I thought I'd give you an example from our own lab where we studied this process for a couple of different polymers. One is this PBAT polymer, which is a mulch film. And PBAT refers to, the P is for poly, but the B is for butane diol, which is this substructure here. A is for adipic acid, which is the 6-carbon diacid, and T is for terephthalic acid, which is this guy here. And yeah, so it's one of these mixed diacid polyesters. And maybe we could just talk briefly about why you would want a mulch film and whether or not it's a good idea. I'm an environmental scientist, and I'm a huge fan of mulch films. I think they're really smart because when you use mulch films, your soil retains water better, so you need less water. You have less erosion from your soil, so your topsoil losses are lower. You're blocking the light physically, so you don't need as much weed control chemicals, so you don't need as many pesticides, herbicides, and it's also kind of a poor man's solar energy. You're absorbing energy from the sun, which makes the soil warmer, which makes it more productive and can extend the growing season. So there's like many benefits to using a mulch film. The bad news is you got to cover these huge pieces of land with plastic, and then you got to do something with that plastic. And as we saw in that example from China, where they use very thin plastic, it gets very hard to recover, and a lot of it gets entrained in the soil. And there's been an observation that soil fertility is decreasing as the plastic in the soil is increasing. So BASF was quite motivated by this. This is their polymer. There are other companies that also use PBAT, but this is the one that they've been working with the most. And they were quite motivated to understand what happens to it. And I have to give them a lot of credit. They basically said, we know it disappears, but we don't know what actually happens to it. And they said, can you figure it out? And we said, oh, of course we can figure it out. I mean, just give us the money, we'll figure it out. And then they gave us the money. We're like, oh no, how do we figure this out? So that was the next step. So let's talk about how we studied colonization. So microscopy was very useful for us. Not only did it give us direct evidence that the films were being colonized, but it also really helped inform us about what was happening in the system. So these are 2 images and 2 blowups of a PBAT film after 6 weeks in soil lab conditions. And this is 25 degrees Celsius. What we see are this network of fungal hyphae across the whole surface. So they're definitely a fungi that are colonizing our material. Excuse me. And if you zoom in, you see all of these really cool little fusilli, these little, corkscrew microbes, unicellular microbes, just lined up like pigs at a trough right along the hyphae. And that was already a hint that maybe the fungi are the drivers, that they're the ones who are doing the main degradation of the polymer. They're the ones that are excreting the enzymes that are breaking down the polymer, and that the unicellular organisms are either taking advantage of the situation and just eating, the substrates as they get liberated, or they might be symbionts where they are taking advantage of it, but they're maybe also providing something else. And we've hypothesized about this. The polymer doesn't have any nitrogen in it, and all the organisms need nitrogen, and maybe some of these guys are nitrogen fixers or something. So we don't know what their relationship is, their biological relationship, but we see them always co-associating with each other. We see that the hyphae can actually burrow holes into the plastic, which is pretty cool. And again, you see unicellular organisms all lined up alongside the hyphae. You can see a couple of these fusilli, but some other more classical rod-shaped guys as well. So that was pretty clear that that colonization is occurring, and that the fungi were really important. And it was one of these things where I'd like to say we had read the literature first, and then we came back later and did the experiment. But we actually did the experiment, and we went and read the literature, and we're like, oh yes, fungi makes lots of sense, because if you have leaf litter in a soil, leaves have a coating on them, which is cutin. It's that waxy coating. You can scrape it off with your thumbnail. And that is a polyester. And the main cutin degrading organisms are fungi. Actually, fungi are probably these are the guys who've sort of specialized in breaking down all of the vascular plants, like, all the trees in the world. And so it made sense later that these cutinases excreted by fungi would be important. lost control here. Let's see if this works. Yep, good. That was in lab conditions. We can also do it with a different polymer. This is PBS, a simpler polymer, where the S is succinic acid, so a 4-carbon diacid. And we did this under field conditions. So we actually went to a real agricultural field and buried the stuff for 6 months. And miracles, we found it later. We found the plastic clips later. And we saw breakdown of the plastic, disintegration of the plastic into microplastic and some larger pieces. We took some of the larger pieces, we looked at them under the microscope, and we saw colonization by fungi and unicellular organisms. We're going to zoom in right here on this T-junction. And what you can see at that T-junction is really good visualization of how these fungal hyphae are excreting these extracellular enzymes and hydrolyzing the polymer. And you get this kind of spongy texture, morphology of the polymer that's right next to the hyphae. Meanwhile, like a couple microns away, it's totally apparently unaffected. And we don't know what the origin of that morphology is, if it's just widening existing gaps that were in the structure, or if it's preferential hydrolysis of certain domains, or what it is, but it's something we see a lot. Okay, so colonization occurs, it's a thing, and we think that the fungi are very important. Next up is depolymerization by extracellular hydrolases. I'm just going to skip through this pretty quickly because I've been just chatting away with myself, and I'm getting behind on time. But we did this 3 different ways. I'm going to show you one of the ways, and I'm going to show it to you relatively quickly. But what we did was we used this molecule as a signaling molecule, as a probe molecule. It's called fluorescein dilaurate. It has these laurate groups on the closed ring form of fluorescein, which is not very fluorescent. But these are esters, and if an esterase comes along and clips these, then you get fluorescein, which is incredibly fluorescent, thus the name. We took this molecule and we buried it in polymer. We basically dissolved polymer and this stuff, and we entombed this molecule in there. It's so large, it doesn't diffuse out very quickly. Basically, on the time scale of the experiment, it doesn't diffuse out at all. And so the only way for an esterase to get to it is for the esterase to eat through the polymer to get to it. So the growth of fluorescence was a proxy for the progress of esterase eating the polymer. This is an artist's representation of what's happening, which, I'm sure it looks exactly like that. And because I'm short on time already, there's other stuff I want to talk about. I'm not short on time yet, but I want to save some time for other stuff. I'll just say that we basically use this to screen a bunch of different polymers that had different chain lengths. And what we found, I think, interestingly, is that short chains were slow, long chains were slow, intermediate chains were fast. And we puzzled over this, and eventually we figured out that the ones with the intermediate chains had the lowest melting temperature. And melting temperature is a proxy for how mobile the chains are. And we think the mobile chains are easier for the esterases to clip. And you get higher melting with short ones and long ones for different reasons, but it's easy to justify why in both cases. Okay, sorry that was so brief, but I want to spend a little bit of time on this last step, this utilization of the polymer. So now you know that the material gets colonized, you know that the ester bonds get broken, but do you know that the substrates that are liberated are taken up by the organism? And to solve this problem, we really leaned on our industrial collaborators. BASF synthesized for us at great expense a whole family of carbon-13 labeled polymers. And we were able to use those to basically track the carbon through the system. So we do 3 kinds of experiments. We use cavity ring down spectroscopy to look at the CO2 being produced during the experiment. And we can distinguish between the CO2 that's coming from the polymer being degraded compared to just the soil carbon, because one's labeled and one's not. Afterwards, we use the label in a isotope ratio mass spec experiment to quantify how much residual carbon was left in the system. That could be residual polymer, or it could be carbon that was converted into biomass. And then we distinguished between those two pools by doing a third experiment where we extracted the sample with deuterated chloroform and did proton NMR in a quantitative way. We're able to quantify how much was polymer. The cavity ring down system looks a little bit like this. We start here. We have an incubator. These two big incubators with lots of bottles, 36 bottles in each, and we flow air over the bottles at all times. It's actually moist air, and it mostly goes to exhaust. So there's a constant stream of moist air going over them. Every once in a while, we divert the air stream into a cavity ring down analyzer, and it's basically a fancy infrared experiment that allows you to distinguish between carbon-13 CO2 and carbon-12 CO2 and quantify them. And we do that experiment, and we do it continuously for a year and a half. So here is PBS labeled in 3 different places. The succinic acid carboxyl positions, the succinic acid alkyl positions, and the butane-diol, all the carbons. And what you see is an initial burst, which I'll explain in a second, and then a slow increase in the mineralization rate, and then a slow decrease in the mineralization rate. If you click cumulative CO2, it looks more like an S shape. The gray curve here is our standard. It's cellulose. Cellulose degrades really quickly. And after about 15 months, we stopped the experiment. We had to stop it at some point. It wasn't done, but you got to graduate at some point. And what this burst is in the first just hours, we see this burst of stuff, and there's small pieces that leach out of the material at the early stage of the experiment. And we've done some mass spec characterization, and they won't surprise the polymer chemist. These are rings. These are small rings typically. They can be also, I think, small oligomers, but they tend to also be little rings of the polymer. And they diffuse out and are apparently very delicious bacteria food or fungal food. Okay, so we know that this is degrading. We're making a lot of CO2, but we also know that there's a lot of stuff left. We're not getting to 100%. And so the question is, what is this remaining carbon? It's still in the system. Is it unreacted polymer or is it biomass? Well, first thing we wanted to do was just see, is it just still there? I mean, we've been running this experiment for 125 days. Please God, let there not have been a leak, in the system. And we were gratified, extremely gratified to see that if you combust the residual material, you can account for essentially every carbon atom, between 98 and 100% of the carbon atoms that were labeled that we put in, we either came out as CO2 and we saw them, or stayed in the soil and we saw them. And that was really wonderful. But we still don't know, is it biomass or is it polymer? And we did this extraction analysis. I have a backup slide I can show you the data, but basically 70% to 100% of that material we could assign to polymer carbon. So almost all of the residual is polymer. That's interesting. So do they just burn it? Do they not incorporate any of the carbon into their biomass? Well, we did one other experiment I wanted to share with you, and this is the last experimental data I'll share with you. We also did the same type of experiment with PBAT, where we had label in the A, label in the B, and label in the T, all at about 4% label. We ran them for 40 days, just long enough to get some microbes on the surface. They actually went at slightly different rates, which was interesting, and we can talk about that if you're interested. And then we ran over to Vienna, where we have collaborators who do nanosims, Mickey Wagner's group, Dagmar Wöbken in particular, who was helping us. And the way nanosims works is you shoot an ion beam, and you basically get a spray, a dust cloud of ions, and you can image those ions using mass spec, and you can make these really, really amazing images that not only look like micrographs, but can tell you what elements are present. So for example, you can look for nitrogen, which should be associated with biomass, and the underlying polymer just looks black. And the hyphae, the unicellular guys, all light up like light bulbs because that's where the nitrogen is. When you look for carbon-13 in particular, you get really interesting results. And we could probably spend 40 minutes just on these results, but I'll try to give you the quick version. If the background is about 4%, we would expect that to be kind of 3 or 4%. It should be, I think it's actually 3.5%. It should be kind of like this, maybe this color is what we expect. What we see when we label the A is it looks a little greener. The surface looks a little greener than we expect, looks a little depleted in A. And the hyphae are red. They're really enriched in the carbon that we had put in A. So it looks like the fungi love to eat adipic acid. Who knew? Now we know what fungi like to eat. It looks like they also like to eat the butanediol, but they hate the terephthalic acid. But that's okay, because other guys, the unicellular guys, seem to love it. And we think this is really fascinating, being able to get almost a biochemical look at what the substrate preferences in real, soil samples in polymer degradation. Okay, so basically that's what we think is the best way to show that material is actually can break down in the environment and can actually be used by microorganisms. I don't think you need to necessarily do the last step, although we were very happy that we did. But I think if you can monitor the CO2 being produced and verify that it's coming from your polymer, that's really the most powerful proof that you have true biodegradation and not just formation of microplastic. Okay, so just one last word about plastic materials. So now you're ready to go to the environment, right? Well, hold on. Plastic materials are complicated. They're mostly polymer, but they're not 100% polymer. They have all kinds of other stuff in them. This is a compilation that was in this Journal of Hazardous Materials review article talking about different polymer additives and different typical weight percentages you might find. Now these vary a lot. Like the polymers we use have no plasticizers in them, but they do have photostabilizers. And the photostabilizers, they would typically be at 1 to 2% of the polymer mass. They have these fillers. Calcium carbonate, I think, is something like 10% of the polymer mass or something like that. So, so this brings up an interesting question. Let's say you have a toxic UV stabilizer. Is it better if it's locked up in a persistent plastic coffin, or is it better for that plastic to be biodegraded and to release that toxic UV stabilizer? And I think you're kind of in a no-win. That's a no-win question. You want a biodegradable plastic and you want a non-toxic stabilizer. But this is something you really have to think about right from the very beginning of designing your material. What I can tell you is our collaborators at BASF, they've taken this to heart. They use primarily food-grade carbon black. These are black gummy bears. So food-grade carbon black as their pigment for their mulch films. Okay, take-home messages. So biodegradability. These are what if you don't look at anything else, please take away these 4 messages. Biodegradability is both a function of the material and the receiving environment. It's a relationship. Plastics that they can disintegrate, they can disappear, and it might look like biodegradation, but it might just be that you're making microplastic. Or it could be it's really biodegrading. And how would you know? You have to look for the CO2, and it has to be attributable to that material. Biodegradable polymers are one part of biodegradable plastic materials. There's also additives. I think this is a huge, important area of research. And finally, I just want to, again, just say how happy we were that somebody actually finally called us and said, hey, do you want to help us figure out if this is sustainable or not? And I think academic industrial partnerships provide unique opportunities. I should maybe also say, be clear that it's also unique challenges for environmental science. And I think it has the potential for improving environmental sustainability because, basically you can provide some input at the early design stage of some of these things. And I think that's very powerful. Lastly, just I want to acknowledge everybody. I did it mostly through the talk. This is a group picture we took 2 years ago, back when we didn't have to wear a mask. I thanked Dagmar and Mickey Wagner and Arno already from Vienna, Thomas Bucheli from Agroscope, the SEM group at University of Zurich, BASF for funding, and also ETH. And I want to thank all of them. and of course to thank all of you as well.